let's kick it off. Welcome, everyone. Welcome to the Caring Cross guest speaker series. We are honored today. We have Trent Spencer from Emory in Atlanta, Georgia. Trent and I were just talking about different ways to stay busy and stay in shape out in Georgia, out in the lakes and whatnot. Today he's going to talk about something incredibly interesting. It's a new approach to hemophilia A. It's a gene therapy approach, and he's just published an impressive paper in New England Journal of Medicine. I'm sure he's going to talk about that today. And Trent's going to talk for about a half an hour, and then we can open it up to conversation. So without further ado, Trent, I'm going to hand this to you and let you share your screen.
I sent a request.
Okay. So.
Okay. Okie dokie. Thank you very much. I think this is coming up now. Okay, Can you see it?
Perfect.
Great, that's step one. Okay, glad to do this. But the one thing I did forget is that this is March Madness time, and I'm a very avid college basketball fan. And I told my wife, I said, this may be the quickest Caring Cross lecture they've had if Alabama starts going down any more than they have. So I'm glad to be here. I'm glad to discuss some of the things that we've been doing in the realm of hemophilia, and especially hemophilia A. As Kevin was mentioning, my background, I'm a professor at Emory University. I'm the director of the Cell and Gene Therapy program within the Athletic Cancer Center. I co-direct the Center for Cellular Therapies in Children's Healthcare of Atlanta. And I am the past president and co-founder of Expression Therapeutics. And I'm the past scientific co-founder of Innate Bio, which is now publicly traded. So those are some of my disclosures for this talk, but I am no longer with either of those companies. So now it's mainly me and Emory that's going to be presenting this. So to get into this, that's when I was asked to give this lecture, I've given this, we've talked about this in worldwide audiences and whatnot, and I've not presented this in the context of how do you actually do what we did when we got this actually through approval process in a country that had never seen a gene therapy approval before. So this was one of the first approvals that went through the Ministry of Medicines within India. And I wanted to touch on some of the things that we had to go through, because I think there's a huge opportunity in the gene therapy world to go into these developing countries and low and middle income countries and actually do some good. And I think when we talk about hemophilia A, I'll mention why this is a passion of mine, and I think it's something that I will continue to do for some years to come, I hope, based on the studies that I'm going to present today. So as you mentioned, a lot of what I'm going to talk about is this New England Journal paper that just came out last month. I think it was originally online back in December or January, but I think it just came out in print about a month ago or so. And this is a project. You can see that I'm the senior author, but this Alok Srivastava, who's the first author, he is the driving force of what happened in this country for developing this gene therapy for hemophilia A. And I want to right at the start give him huge kudos, because without him, none of this was possible. And I'm gonna harp on that again later. When you actually try and do this, you have to have somebody like Alok bringing this forward and doing the yeoman's lifting to get this done. But there's also people like Chris Doring, and who's a good friend and a colleague of mine, who helped develop all of the gene therapy stuff around this program, and Pete Lawler. A lot of the discoveries I'm going to talk about were from Pete. And then Gabriella Denning did all the regulatory work that was needed. So there's a lot of thanks to go around. And I wanted to show some of these names at the beginning to get that out of the way in case we run out of time or something. So this is kind of the foundation that I'm going to be working toward, is this results that we got in India, but we're going to discuss more than that. So at first, I wanted to touch on where this came from, and this is a gene therapy using bone marrow transplant that has highly technical strategy, and I'm going to talk about the technology in a little bit. But this technology was developed at Emory and Aflac, and a group of us put this technology together. When we did this and we tried to move it forward, it was clear we needed a company to go out and fund this at higher levels than what we could through academia. So we developed a company called Expression Therapeutics, and then Expression Therapeutics at the time didn't have vector manufacturing. And I don't know if Boro is online, but I thought if he is, I wanted to give him a shout out, because from the very beginning, Expression partnered with the company called Virexis. And this is way back when Boro was starting out in his company ventures. And we were talking to Virexis and partnering to try and move a gene therapy for hemophilia forward using lentiviral vectors. He was the natural person to contacted him. We did a lot of translational development with Virexis. But then, as we know, Virexis Boro developed Lentigen, and then Lentigen went elsewhere. And when they did that, they weren't as interested in developing a gene therapy for hemophilia. So we were kind of left on our own to try and develop this. So what we ended up doing is I went up and I talked to a bunch of the other vector manufacturers within the U.S. and came across Bill Swaney at Cincinnati Children's. Bill is a great guy. I give him tons of credit for taking this on as a project within Cincinnati Children's to make this vector for us. So Bill ended up making this vector, and then eventually, I'm going to make a long story short, Bill ended up joining Expression Therapeutics. We thought that highly of him. So he came, joined Expression Therapeutics, and we've done a lot of vector technology and a lot of vector manufacturing and a lot of additional technology development around this program. So from the start, and I'm talking 15, 20 years ago, where we were working with Boro, and this concept was formulated long ago. But then as we started bringing in these people like Boro, like Bill Swaney, the Chris Dorings, the Pete Lawlers, Gabriella Dennings, these are great people that were doing great work. And then we were able to bring them together for this project. And I think that's one of the foundational aspects that allowed us to be so successful. Is from the beginning, we were working with the best people in the industry. And that was very rewarding all along. When I look back, I don't know how fortunate I was to be able to find the people that we actually worked with. So I'm very grateful for all the people that helped us along the way. So now I'm going to get into some of this technology development for this project. And now we're going to start talking about the nuts and bolts of what we're doing. So hemophilia A, just real brief background. Hemophilia A, it's a deficiency in factor VIII, which is in that star. You have, it's an X-linked monogenic disease. Factor VIII is mutated or deleted. So there's a lot of things that cause factor VIII to be missing or non-functional. So if you don't have factor VIII, you don't finalize the clotting cascade, and you don't clot well. And then the people with hemophilia A can have bleeds, and you usually think of them as maybe cutting themselves with a knife, and that's problematic. But really, that's not the problem. It's walking across the room where you have these microbleeds, and you just can't fix all of these microbleeds throughout your lifetime. You end up with arthritis and brain bleeds, and you eventually succumb to the disease because of bleeding in these locations that are life-threatening. So the treatment of hemophilia A, the standard of care for hemophilia A is to inject 2 to 3 times a week with factor VIII protein. And that's been the mainstay of hemophilia treatment. Now, that protein can be recombinant. It can be isolated from blood. In the past, you could take your neighbor's blood and inject that into you to try to alleviate it. We've certainly come so much further than that. I don't have time to go to the history of hemophilia A treatment, but now we have recombinant proteins. We have bispecifics that are working well. So there's a lot of treatment advances within hemophilia A. But in this case, what we're talking about, and I don't know if what we're talking about is that protein replacement, which is the standard of care. Protein replacement is extremely expensive. It's $300,000 or more per year. And if you look at this cost and you look at factor VIII usage, Because of this cost, there's this huge treatment disparity. Where in the US, the UK, we're using substantial amounts of factor. But then as you go down to these low and middle income countries, which I'll refer to as LMICs or low and middle income countries, you can see that factor VIII usage is almost non-existent in a lot of these countries. And the reason for that is that it's so expensive. So there's a need to alleviate this treatment disparity. And that was one thing that Expression Therapeutics was founded on, trying to identify ways of treating low and middle income countries. And this became a major theme of the company for a while, to try and get into low and middle income countries because of this cost. But not only that, it's hard. You have hemophilia from birth. You have 3-year-olds, 5-year-olds needing factor VIII injections. And these kids, they learn to inject themselves 2-3 times a week. It's really amazing to watch a little, almost toddler age, starting to inject themselves. So it's invasive. It's hard. It's very strenuous to keep up with what would be considered standard of care. The accessibility, because of the cost, only 30% of the people on the planet actually get standard of care. So 75% of the people in the world aren't treated well for hemophilia. So there's accessibility issues. And then 30% of the people that are treated well actually develop inhibitors to the protein product. So because you've never seen this protein before, it's actually immunogenic to a lot of people with hemophilia. So that's another strike against it. Now, all of this together, gene therapy, as this group knows well, there's been a lot of effort focused on gene therapy for hemophilia. Now, the FDA has approved an AAV factor VIII product. I'm not going to go into AAV bashing for factor VIII, but the product there's issues with it. It's not a lifelong cure. The factor VIII levels diminish over time. There's problems with the delivery. There's problems with the variation of how much factor is being expressed by each individual that's injected with this product. So there's concerns about the product, but nevertheless, it's an FDA-approved product for AAV. That said, and everything else that's going on, we've thought that there's opportunity to improve upon this treatment modality. Also, the FDA approval of AAV factor VIII product, this is in the millions of dollars, and it's equally expensive treatment. So this is not alleviating any of that treatment disparity that we've seen in the history of treating hemophilia. So with that kind of as background, this is what we set out to do. We set out to cure hemophilia. In fact, we sat down with the treaters at Emory and said, okay, if you could develop a treatment using a gene therapy that may not be lifelong, say AAV at the time, this was a couple decades ago or 15 plus years ago, where AAV wasn't integrating. We didn't know how long it would last, but we assumed that it wasn't going to be lifelong. Or if you could develop a lifelong treatment, which would be your lentiviral vectors because they integrate. And if you could use a stem cell that was highly characterized, which would be the hematopoietic stem cell, which would be the better path to go down. And it was fairly unanimous from our treaters that they'd rather work on a lifelong treatment. So that's why we came up with this. Now, basically, everybody knows this paradigm where you have your recombinant lentiviral vector, you harvest hematopoietic stem cells, you transduce those ex vivo, and then those gene-modified cells are injected back into the patient. So that's the concept and modality that we're using. Now, there's lots of pros and cons to this. One is the pros is that there is extensive experience using lentiviral vectors, and I think the more data that's been built on this, I think the safety is just being built and built, and I think the safety is not something that we need to pontificate about. I think it's a reasonably safe way to go about treating and transducing hematopoietic stem cells. That said, we can have that debate, and I'd gladly debate anyone over those issues. Age is not a barrier. You can treat young kids. We transplant at the Aflac Cancer Center. You can transplant babies that might have cancer or some other diseases. So age isn't an issue where it is with AAV. And then you don't have anti-AAV antibodies. There's a large population of people that can't be given that AAV treatment because of anti-AAV antibodies. But the con is the complexity and highly involved process. And this is important. When you're thinking about the complexity of doing this, where you have the lenti production, you have the cells, even thinking about this it's highly involved. So doing it in the US is problematic, but then thinking that we could do this in a country that's never done it before, that was a hurdle that we all had to get over. It's like, can this actually be done in a place like India that's never done this sort of gene therapy before? So the complexity is one of the major hurdles. Now, what I'm going to talk about now is the technology development. Just real fast, I just wanted to talk because the technology is what drove this program. We needed the technology advancements to actually have this work. And the discovery was by Pete Lawler, who was identifying ways to overcome inhibitors to human factor VIII. And one way to do that is to give porcine factor VIII. If you have inhibitors to human, you can give them porcine. In the course of those studies, he had the seminal discovery where porcine factor VIII is expressed at 10 to 100-fold higher levels than human factor VIII. So that was incredible. And that was something that we found, that high level expression. We started using porcine sequence, and it was like turning a switch on. Things that didn't work now work like cutting butter with a hot knife. It really changed the trajectory of these studies. But then over the years, Chris Doring and some other people that were working within this program went and identified the residues within porcine factor VIII that were required for this high-level expression. And then they transferred those sequences into human and made this human porcine chimera that we called ET3. So ET3 is a human factor VIII that has the relevant porcine sequences for high-level expression. We then tested this in vitro settings, in vitro and in vivo, and we showed that the ET3 construct was very similar to the porcine construct and had very high level of factor VIII expression. So from that, we also identified one of the mechanisms is this lack of inducing of the unfolded protein response. When you express human factor VIII, it was one of the proteins that was actually identified, the unfolded protein response. You express high levels of human factor VIII, the cells, it gets mucked up in the cells, and the cells die from this unfolded protein response. Porcine factor VIII and ET3 do not induce the unfolded protein response similar levels as human factor VIII. So this was an advancement as well, and something that was very exciting. So then we took this on to gene therapy platforms. My background is with Art Nienhuis and Brian Sorrentino. We were at St. Jude Children's Research Hospital. I think Art Nienhuis is one of the fathers of gene therapy that was driving a lot of the lenti and retroviral development at the time. And I had a ton of experience with lentiviral vectors. So we started on that platform because we could do AAV. We have everything in the lab, AAV, lenti, all the different serotypes and whatnot. But we went with lenti for the obvious reasons that I'm going to talk about. So I made these constructs with the factor VIII transgene. And actually working with Bora, we found that some of the stronger promoters and some of the ubiquitous promoters just didn't make vector very well. We had very, very low titers. And it was all we have a couple papers on this, and it was because of the promoters that we were using that were they were actually stealing transcription factors from the LTRs and driving the mRNA for the transgene instead of the packaging RNA. So we did a lot of manipulations. We tested a lot of different promoters, and we came up with this construct, which had a CD68 promoter driving the ET3 transgene. We made lentiviral vectors. We took that lentiviral vector, we transduced hematopoietic stem cells, and then from this, using that promoter, expression of the transgene is driven away from the hematopoietic stem cell and progenitors and away from the lymphocytes and mainly expressed in the monocyte lineage. So we have this, what we know is a safety enhancement for factor VIII expression. We also had data showing that lymphocytes do not make factor very well. They don't express factor that gets built up in the cell. So this was a way to overcome that discovery as well. So also when we do this in a mouse transplant model, this is tolerogenic in hemophilia A mouse models. So now we have a model, we have a construct. we started doing our preclinical development and in vivo IND enabling studies where we leaned on the hemophilia A
Trent,
mice.
can I jump in for a second?
Yeah.
I've got a question, if you don't mind going back. Was there any natural endogenous expression of factor VIII in those cells with the blue arrow? Is this all exogenous?
So factor VIII is typically expressed from liver endothelial cells. So this is driving expression away from its natural expressing cell types. So it is the liver endothelial cells. So the answer is not to our knowledge that there isn't factor coming from these cells. You'll see the odd paper here and there where you may be referring to, where you see some of these expression, these papers that might show that a transplant might be curative for hemophilia A, but in our hands, we never saw that. Clinically, you don't really see that either. So the answer is that these cells don't typically express factor VIII, so there's no endogenous factor VIII being expressed either.
All right. Thank you. Interesting.
Okay. So then we went to the hemophilia A mouse model. We leaned on this heavily, showing the concept is solid, that we could get this treatment modality to work in hemophilia A mouse model. Then we used the NSG mouse model to transplant human transduced bone marrow cells into an NSG mouse, and we showed that we don't see any adverse effects with this transgene and this specific promoter. So from that, I'm not going to show all of that data, but we had this data where you can look at the vector copy number within their peripheral blood mononuclear cells, and you look at factor VIII activity, and we saw this dose response. The more vector you have that engrafted in these animals, the more factor that you would express, and you can get into the normal range of factor VIII. So using this construct, using this transduction platform, and using this assay, we could say at the time that using technology that was readily available to get copy numbers in the 0.5 to 1 range, we thought it was a go because we knew we could achieve that in humans. So we went forward with this and went to the FDA. We submitted a pre-IND, and then we did the IND that was submitted in 2019, and then the US FDA actually approved our IND in 2019. We had meetings with key opinion leaders and hemophilia A patient advocacy groups, and the drug product was made by Bill Swaney. We had that vector produced. If you look at this time, 2019, the problem is that the pandemic started soon after, and Emory shut down their clinical trials, and so did everyone else. So this was put on the back burner, and this was a terrible time because we had everything lined up to do this study. But as luck would have it, Chris and I had made a couple trips to India because this is one of our passions, is to try and get LMICs to try to treat low and middle income countries. And as I said, that was one of the pillars that Expression Therapeutics was founded on. So we were going into India, and one of the questions we originally had is, well, is it really as bad as we think it is? And you read a paper and you see, and then you think, okay, well, what is it really like? So we had boots on the ground. We started talking to people, and it was bad. The treaters there are awesome. They did the best they could with what they had. The people with hemophilia are awesome because they know they have hemophilia. They have to live their lives in a way that they don't damage their joints and whatnot. So they all know what to do, and they manage the disease as well as they can. But the cost just prohibited the treatment there. So we were trying to get some low-cost treatments into India. And one of the things, so we went to India several times. This is us traveling with Mohan Rao, who's the CEO of Expression Therapeutics. We went there, saw that it was bad. But one of the things that came out of this is this guy, Alok Srivastava, which I picture in the bottom right. Alok Srivastava is at CMC Vellore. CMC Vellore is where that red dot is in the top left. And CMC Vellore is pictured in the top right. And what we found is that Alok Srivastava ran a top-notch, wonderful cell therapy GMP facility. This is in Vellore, India, which is between Bangalore, which you've probably heard of, and Chennai. This is you travel 2 hours from each of 4 hours from each of those into India. You come into Vellore, which is a fairly, they call it a small village, but it's probably several million people. But so you go into this place, and you would never imagine that they had this GMP facility in the stem cell research program that Alok was heading. Alok was very interested in the gene therapy program because he knew that if you could use gene therapy, this would be curative. He had hoped, and he thought, this is what we need. We need to be able to treat these people early, so that the earlier you treat them, the less problems they're going to have that's been pre-established before treating. We need a treatment that's going to be early, able to be used early, and effective. So he was already world known for a lot of things that they were doing in hemophilia because he was ingrained in hemophilia in India. So it was by happenstance that he's also a bone marrow transplanter. And here we are doing hemophilia using bone marrow transplant. So he could understand, and we could talk the same language for this program. So Alok, after talking to him, convinced him showed him the data was convincing. Then we had our IND approval, and we were going through the IND process. So he's like, well, why can't we do that here? So after several meetings and after several trips into India and visiting and doing our due diligence and whatnot, we thought, why not do it in India? And when we think back about it, the better question is why? And there's so many reasons not to do it in India. And mainly because it's never been done. Nobody had made vector before. Nobody's gone through the India FDA to get something like this approved. So we knew that, but those of us who haven't done it think we can do just about anything. And we thought, well, those are hurdles. Let's try and start getting through them. So we did. This is what we thought would happen. We had our US IND, and we thought, okay, we've got our farm tox. We'll just put our farm tox into the India regulatory programs. We had our CMC. We put that into the India. The protocol I'm going to talk about remodified, and the modification was such that in the US, we wanted to do reduced intensity because there was treatment, and we thought an ablative transplant was not the way to go because reduced intensity seemed like it. There's a lot of discussion about the protocol. In India, Alok was like, look, an ablative transplant would make sure that this works. If it doesn't work in an ablative, it's not going to work anywhere else. I want to start high and go low. You guys can start low and go high, but I want to make sure my patients get the benefit that they can achieve from this if it's going to work. So they want to do an ablative transplant. So we said, okay, we rewrote the protocol and we submitted that. And we went through all this filing from 2017 to 2022. It took us 5 years. We had the US IND pretty much in hand. It took us another 5 years to get India to actually approve this. Even though we had, anyone who's put in an IND, the investigator brochure is something you don't write up front. You kind of put that together as you're doing it, and the final protocol, you kind of put it together, submit it to the IRB and that sort of thing. 2018, we had the investigator brochure done. So I just mentioned we didn't have this trial going until 2022, approved till 2022. And the reason is because we had to go through all this. Now I blur out all of the committees that we went through because I don't want to badmouth any of the committees that we had to meet with because they did as well as they could do. The problem is that nobody had the authority to allow us to do a gene therapy clinical trial in India. So we started going through this in about 2017. We had the brochure in 2018. We were going through all the different committees within the Indian medical program to try and get this approved. And every week we'd go through somebody else, and every week they'd come back with more questions. And every week we were answering questions, and this went on for years. And then finally we're like, we either got to do it or we don't. And then in 2019, as all of this was going on, they realized that the government proposed the creation of this independent body for biomedical and gene therapy, and that was called the Gene Therapy and Advisory and Evaluation Committee, GTEAC. Now, when you look at this bottom, I don't blur out GATC because when we went to the GTEAC, that was the final. They said, okay, we want you to do some of these things. There's a few things that we need. And then once you do that, you'll have your approval. We did it. We got our approval. So that was a long long road because it had never been done before. Now, when you do this, there's at least a pathway that people can take, but we had to to navigate that pathway. So this is the study that was actually approved. It's called the primary objective was to assess the safety and feasibility of doing this in hemophilia subjects because it had never been done before. And then secondary is does it work? So, but this was a safety study, obviously. So we went through the safety study using CD68 ET3. CD68 is a promoter, ET3 is a transgene, lentiviral vector transducing hematopoietic stem cells or CD34 cells. So that was the study. Now, the other thing, we had made the vector in the US, so we had to ship that off to India. So we shipped a bunch of the vector, hundreds of thousands of dollars worth of vector to CMC Valor. We're all on the edge of our seats. We tried to ship it. It didn't go through. We couldn't get it shipped. And the problem is the customs between the US shipping a lentiviral vector at that time was very hard to get approval for because we didn't know who should give us approval. We finally, and COVID was going on. So we were trying to ship this where flights weren't flying. We couldn't get this through customs, and it was sitting. One thing that we did learn is lentiviral vector is very hardy. You can put it on -8 in liquid ice. You can put it in several different actual things, and it's very sturdy. It's very sturdy. So we were able to ship this. We finally got that worked out. We shipped it, and everything worked out because once it got there, they actually repeated the titering. They did everything that we did to qualify it, and it passed with flying colors. So this is us actually shipping the vector and that's the van driving off with our hundreds of thousands of dollars of vector to unknown parts that's never seen this before. So then this is important on the differences between the US and India. In India, they don't use busulfan. They don't use, well, they do, but Lok in his institution used triosulfan. Triosulfan, similar to Bu, but it doesn't have the side effects. Trio just got approved in the US, by the way. And this is one of the things that we were pushing because we had such good results with Trio in this trial that I think it should replace Trio and Melphalan and everything else. But that's my opinion from this one trial with hemophilia A subjects. So we had an ablative amount of Trio, we had fludarabine, and then we had autologous transduced HSCs. Stem cells were mobilized stem cells. And then we mobilized enough to inject at least 2 × 10⁶ per kilo. And the minimum was, I think, 5 × 10⁶ was the minimum dose that these subjects received. And then we had an aliquot of unmodified CD34 cells that we kept in case something happened. So then this is the processing. It's the natural flow. There's a couple things I wanted to bring out on this processing. We did all the routine analysis of this, but this is important to understand for some of the later data. So initially what we did was we identified the way to transduce hematopoietic stem cells here at Emory and within Expression Therapeutics, and Gabriella Denning developed wonderful transduction processes that had fairly high level transduction, but it was a 2-hit, 2 administrations of vector over about 24 hours. And that worked well. But then, as I'm going to show, we thought we could do better. So we switched from that and we said, okay, if we want higher copy numbers, we could use an enhancer. And I'm going to talk about the enhancer. And as it turned out with the enhancer, we only needed to use half the vector and do only a 1-step transduction. So then in the first couple subjects I'm going to show, they were 2-hit transductions, and the later ones used a single transduction step. Also, when we did this, we did a CFU assay where we plated the bone marrow, transduced bone marrow, in methylcellulose and let that grow. And those colonies were used to isolate DNA and actually look at transduction efficiency prior to the transplant. And we set a cutoff of 0.5 copies in this assay before we actually felt comfortable transplanting the cells. So this is the first subject. These are the patient characteristics and the post-engraftment. There's nothing phenomenal about this. It was very routine. If you look at some of the important numbers is that these subjects had factor coverage for about 2 weeks after the transplant. So we continued to treat these subjects for about 2 weeks after transplant. After that, we started tapering down, and then you're crossing your fingers at that point because you never know what's going to happen at that time as you start tapering down factor levels. So these are our first 2 subjects, and it was exciting, but it was a little disappointing. And the reason is because the factor VIII levels were low, and this is participants 1 and 3. So actually, this is our first 2. Participant 2 had an inhibitor and was eliminated from the study. And as you see, I think we have participants 1, 3, 4, and 5 didn't meet inclusion criteria, and then 6, 7, and 8 were the ones that we treated. So we treated 5 total, 2 with this first protocol. You can see that the factor levels are low. So, but they weren't bleeding. So we thought, okay, this is good. Safety was shown. These subjects did very well. Let's try and improve our transduction efficiency. So what we did was we simply added an enhancer, and the enhancer we used was Lenti-boost. And the reason we did Lenti-boost because it's GMP qualified. You can order it. They've already GMP qualified the product. A lot of these other ones that work well, as I show here, you can get you can go from 0.5, 2.5, 5-fold increase in VCN just by adding these different treatment regimens. But not all of them are clinically available. So Lenti-boost we talked to the company, they worked with us very, very well, very nicely, and we were able to incorporate Lenti-boost into the transductions. So then if we look at the next 2 subjects, which would be 3 and 4, participants 3 and 4, you can see that if you focus on the VCN at day, 30 after transplant, see the first 2 had fairly low copy numbers after transplant, 0.07 and 0.12, which would be about 7% and 10% transduction efficiencies, which were low. But then when we use Lenti-boost with a single transduction, we have copy numbers of 2.5 and over 1. So we had high levels, and remember that 1.45 for that because it's going to come up on the next couple slides. So the Lenti-boost worked. We got higher VCNs. So then what happened with the transplant? Well, here's the first 2. And then we got copy numbers around 3. You can see the gray bars, the gray dots, and see that we started getting very, very high levels of factor VIII in the subject. So that that told us that, yes, like the animal model, more vector is better. And then if you look at the next subject, which is in yellow, which had a lower copy number, it was lower than subject 3, so subject 4, but we were still in the 30-40% range, which was still wonderful expression. And then if we look at the last subject that we treated with this regimen, this is the blue, you can see that it had fairly high levels also. When you look at these levels, you can see that the range is now getting into the normal range. So we were able to achieve normal factor levels, and these levels are steady. Once they plateaued like this, these numbers have been steady for this amount of time since this transplant. And I don't have that how long this has been, but it's been over a year for a couple of these. So it's a very, very robust expression of factor. Not only is it expressed well, it works because it happens that 2 of these subjects were in automobile accidents and had lacerations from the accident. This would have been a major problem if these were not treated, if these didn't go through the gene therapy, but they did, and they didn't need factor coverage after these accidents. And one actually had a cat bite as well. So these are things that you just don't do when you have hemophilia in India, but they had lacerations, and they came through these motor accidents just fine. So then if we look at the gold standard, it's like, okay, they bled before. The annual bleeding rate and the annual adjusted bleeding rates are the 2 numbers that you think about when you're dealing with hemophilia. And here, when this first subject had lots of bleeds, it's 30, 26. Those are annual bleeding rates. It's they're bleeding, and after gene therapy, this has been several years now. It's 0. So we went from an average bleed that was typical for hemophilia down to 0. This was the low level expressing subject. They didn't express a lot, but they're not bleeding under these normal conditions. The next subject didn't bleed as much, but after gene therapy, again, 0 bleeds after gene therapy. And again, this was the lowest factor VIII level. We're looking at 3%, 5 plus or minus 2%, because we're down at the lower level of ability to measure factor. So we're at 0. The next subject had lots of bleeds after gene therapy with 0. Next subject had a ton of bleeds. This is what's reported. This is one thing about annual bleed rates and annual adjusted bleed rates, is that this is a reported number. This guy was basically using prophylaxis to cover, if you look at the number of bleeds. But again, lots of bleeds down to 0. And then the last subject, same story, down to 0 after gene therapy. So this was exciting. Everything looked good. If you then look at some of the details, you can see that, like the mouse model, if you look at factor levels versus vector copy number, you can see that it's correlative. The more copies you have, the better factor expression we saw. And this was done, these numbers, what really is striking is that these numbers were done by several different laboratories. The vector copy number were done at in a laboratory setting. The factor VIII was done by the clinical labs. So these are different. It's not like in my lab where the same person will do the activity and then the copy number. These are different groups doing these numbers, and then I'm putting them together, and then this is what popped out. So amazing that you get this correlation. This is what I'm so excited about, and this is what I'm really trying to nail down in the phase 2, is that if you look at the vector copy number of that methylcellulose before the transplant, you can transduce these cells, grow them in methylcellulose, and then get a copy number. That copy number appears to be correlative with factor levels at day 60. So you could potentially tell a participant or a patient that, look, we mobilized, we transduced, you've got a copy number of, say, 1 or 2 in the cells that we're going to transplant. We think that you're going to get 40% factor after the transplant, and I think we can get to that if we expand on this in the phase 2 testing. So before transplant, I think we're going to be able to predict how much factor. It's noisy right now, but I think there's reasons for that. So for this, I think that a copy number of about 3 is achievable. I set that as the target. We could set it as 1. We could set it as 3 because nobody's bleeding. We don't know what the minimum target is, but we do know that these are doing well. And I think 3 is very easy. It's very easy to achieve. We can do it at a vector dose that is feasible as well. But there is room for improvement because, as you see in the mouse actually plateaued, and we're not to that plateau yet, but I think we're in this range where we're getting close to the plateau. So we can go up, but that would require additional copies. And I don't think that bang for that buck is worth the improvement. I think we're in a sweet spot. I think that we can work in this realm that we're in now. So then we did insertion site analysis. This is what I call my icicle plot, where you have each chromosome along the top hanging down, and then each of these dots is an integration in that chromosome. This is with participant 1. And I'm not going to go through insertion site analysis. I hope that most people understand this. The circles are things that we're looking at. We want to follow the ones that are circle, which might be insertion into an oncogene or something that has been shown to be problematic in the past. And then this is with the peripheral blood. And if we compare that to the bone marrow, you can see that the integration patterns are very, very similar. This is without the enhancer. I don't know, Kevin, is this blocking you, or are these pictures blocking you, I assume?
I can see great.
Okay. So then if you look at the enhancer, say this is participant 6, you can see that participant 6 is participant 4, the 4th subject. You can see that it's similar. It looks similar, but you just have a lot more dots. And you have a lot more dots because you have a lot more integrations. As we showed, it was 4 times the amount of integrations. And if you multiply this top by 4, you do each one of these and do 4, this is what you get. So it is what you would expect to have more integrations. Now, when we look at this, this can be a problem. The more integrations, it's more possibilities of something bad going on. But if you look at some of this analysis, if you look at the distance to the transcription start site, you see that in each of these participants, you get about 10% of your integrations near that 10 to 12%. Whether you're doing without enhancer or with enhancer, you see those percentages are about the same. And that's the same distance to CPGs, less than 1 kb from CPGs. You see about 1 to 2%, and that's what you get with the enhancer too. So it seems like they're integrating. You're just getting a lot more integration, but they're still integrating in the genome in a similar locations, which is away from the transcription start sites and away from the CPGs. So that was interesting. The other thing is that if you look at this, if you think about each chromosome and integration within each chromosome, if there was insertion site mutagenesis, one of these dots would be bigger than the rest. And we're not seeing that. So at this point, we're not seeing clonal dominance at the stem cell level within the peripheral blood mononuclear cells or bone marrow analyses. The other thing is that we did a bone marrow examination, and it was performed at 4 to 6 weeks after gene therapy for our participants, and it showed normal cellularity and morphology. So the bone marrow is normal in cellularity and morphology. And if you look at the karyotype analysis was also normal. So far, so good in the insertion of these transgenes. So then if we want to have just a conclusion for this, for these studies, which I did go over, the enhanced factor VIII secretion was needed. The ET3 made this successful, so I think high-level expression is needed. India has this strong interest in doing clinical testing of gene therapies, and then and it was great. They were great collaborators to work with, especially the institution that we found, and you had that person, Alok Srivastava, willing to take this on and work hand in hand with me to try and bring this to fruition. Triosulfan was very good in conditioning hemophilia A participants, and it's well tolerated, and engraftment was uneventful. And then ET3-based gene therapy can provide a functional cure, at least for this long, for hemophilia A. Nobody's bled yet, so hopefully we have this functional cure. And then transgene cassette design is very important because we had to design this around the human and porcine. We had to use the CD68 promoter. It was a heavily engineered cassette. So that's that conclusion. So now what's happening in the real world over there? You can look at some of the newspapers, the Hindu, and see that there's a big write-up, and this is a major newspaper in South India. They had this write-up that Indian scientists developed this novel gene therapy to treat hemophilia A, and these scientists are wonderful, as they are. We have the Prime Minister of India, Prime Minister Modi, is pictured with this analysis or this write-up of this study, where they say Indian scientists make history, and they're talking about this New England Journal paper and the studies that we presented. So that's going on there. In the U.S., we see that the Glenn Pierce, who is the World Federation of Hemophilia Vice President, he has accolades on this, even though I've known Glenn for a long time, and he was skeptical, as a lot of us were, but even Glenn said nice things about it. And then the World Federation talks about the New England Journal of Medicine featuring groundbreaking gene therapy. That's all good. So we got press around the world. It's in the U.S. It's looking like people are agreeing that this is a good gene therapy to move forward. But then if we look at India again and what's going on in India, and a lot of us are interested in CAR Ts and whatnot, where there's another group that was in Mumbai at the same time that we were. They knew what we were doing. We knew what they were doing. And they were trying to develop a CAR T. They were going through all the same struggles that we were at the same time. So we can talk to them and say, hey, look, what are you guys getting? And this is what they're making us do. What are they making you do? And so they made through, and they treated. They're using CAR T, and the reason that they're doing it is they're trying to make the world's most affordable CAR T therapy. They've got it down. It's roughly $30,000 is what I've been told, and then they're trying to bring that down to $20,000, and then the accessibility and affordability of CAR T. So this is a company. This is called Next Gen, I think next car. Yeah, next car 19. So this another company in India, low hanging fruit, CAR 19, CAR T cells targeting CD19. So and it's there, and it's got a company backing, and it's moving forward. In fact, I think it's been approved in that country. So then finally, people that's helped, we've had a ton of people. As I mentioned, I tried to mention the key leaders original in this, and couldn't do anything on our own, and certainly not something when you go into a country like India for the first time trying to do this. So with that, I'll wrap up and open it up. And if we have questions, sorry it took me longer than I thought, but Kevin said it would. So you could have stopped me if you wanted to. So that's our story, and hopefully there's more good to come.
Trent, what an incredible story. Thank you for sharing that. That was highly engaging, and you took us on the whole journey there, an international journey across many, many years. So yeah, first, congrats on the results. I think that's really exciting. I definitely have a couple questions that that we'll get into, and then I encourage anyone. We've got a good audience here today, so jump in on the chats section and type out a question, and then I can read that out. Well, let's see. Well, first of all, since you ended by comparing to CAR T and how India could bring an affordable CAR T forward in that 30 to 40K price, do you think that's in the realm of possibility if this were to go forward commercially in India? Do you think that this could also be in that 30, 40K commercial price range?
I think it's just a little bit higher than that, but not much, because this has to go through a bone marrow transplant, which adds another maybe 10K to that. But we're looking at trying to do it in that same ballpark anyway. So we're trying to keep that number in mind. And to do that, it's a good question, because even in India, okay, it's 2 million here. We can do it. We can cure hemophilia. Let's say 2 million here, 50,000 in India. So let's just say that's reality. And it's like, we can cure hemophilia 50,000. In India, that might as well be $4 million, because $50,000 is still a lot of money when you're looking at the LMICs. So there still needs to be effort in bringing this even within that context. And we're doing that. You have to have a public-private partnership to make this work. And we're doing that. We have the private part, the public part. I wish I had this announcement that was made in India. I think in the last couple of days, they're putting in 5,000 crores into this, which is nearly $1 billion or so into cell and gene therapies. So I think the stars are lining up to get it to work. I think that we can do it, but even then, in this group that I've talked to before, a great group. They were so strong as well. They really put their best efforts forward also to get this product approved. We give them all the kudos as well, and I think we're equally beaten up. And I think that the cost is still a problem. But you can't do it for free. Who's going to do it for free? But then there are places that care about it, and I think that we can do it. It's going to be hard. It's the next hurdle, but I think we've got a group together that can do it if we can bring it forward.
Okay,
maybe Caring Cross can do it.
Yeah, that's definitely in our wheelhouse.
Let's talk.
Okay. Well, first of all, I have a question about, in terms of durability. I'm just thinking, I guess, just kind of brainstorming with you talked earlier about how the AAV approach has led, there's been a loss of durability, and you can think of that in the context of the AAV being episomal and then diluting out over time in the liver cells. This is my naivete on the subject. When I think about that, I'm thinking, is it because of the dilution of the AAV episome, or are there some kind of transcriptional repressors that are just shutting down expression? Is that a contributing factor? And because then when I think about what you're doing with LV, well, you're integrating, so you're eliminating the idea that the episome would dilute out over time. But is there the chance that somehow the transcription could repress on in the 3 to 4 year market? How do you think about this?
AAV? Yeah, with AAV, the other thing, the reason I brought up the unfolded protein response, because that could be happening as well. And you're just eliminating cells that have that's actually making the protein. So the unfolded protein response. Now, there's been a lot of effort in that. And there's some data showing that, yes, the unfolded protein response is triggered, and some data showing it's not that big of issue. The other thing I think is when you start looking at actually what's transferred, because with AAV, this is a huge gene. That's why it's expressed so low. This barely fits in AAV, even when you have the smallest promoter that you can engineer and design the smallest promoters to actually make that work. But there's a lot of different things that are transferred. Pieces of this protein are showing up, pieces of the transgene are showing up, and all sorts of other concerns about that. So there's a lot of reasons why factor VIII may be shutting down, not just the loss of the viral sequence, the viral vector sequence. So I think that's just one thing. There's other things like you mentioned, and then there's unfolded protein response on top of that. So it's being teased out now, and they're working through those. But the other problem is that if you look at, say, an Indian population, Alok and Arun Srivastava, I think it was Arun that worked with them, showed that like 90% of Indians actually have pre-existing immunity to these AAVs, to almost all serotypes. So it's prevalent, it's endemic. So it's really a struggle to find an AAV that's actually going to get over that hurdle. So that's a problem. And then also with hemophilia, the earlier you treat, the better. If your liver is expanding, you're definitely going to get the dilution like you're talking about. And it's more of a concern. The dilution is more of a concern the younger you go. You go into a 2-year-old where you really want to do this, and AAV isn't going to be very good. On the other hand, young children actually go through transplant fairly well. They may have there's other concerns around it, which we can get into, but we think that this could be used earlier also.
Okay, interesting. All right. Well, I have another question. In terms of in terms of safety, so you talked about the integration and how that looks not like it's not a problem, just based on the work there. With the ET3 element, you said it's porcine, it's derived from a pig, right?
Yeah,
so
did you 10%.
See any 10? So a small percent. So it looks like you didn't see any immune rejection based on that. Is that true?
So we did not. And that's why I think that it's important to do this ablative study, because that was one reason why Alok and their team chose to do ablative, was because if you do ablative, you actually reset the immune system using transplant. So the immune system is reset. So we wouldn't, if we saw it here, that would be that wouldn't bode well for trying to titer back on the conditioning regimen, but we didn't see any, and you would expect to. We've done a lot of AAV work and with hemophilia A and whatnot, and I think Chris Doring was the lead author on a really great manuscript that I don't think is getting enough press. Is the speed of factor VIII expression after transplant. If you have a really fast expression and high expression, you're going to get inhibitors developed. And I bet you would here too, if you really drive the expression high and quickly. If you get this high level expression over the first couple of weeks to month, that's a problem. If you see ours, factor levels are going up over time, and then they're peaking, which is another sweet spot of factor VIII expression. So I think we've hit that window of expression as well.
Yeah. Okay. Well, thanks for that. We're at the 1 o'clock mark in Pacific time here. You're 3 hours ahead. How about we end it by just asking what's next? This is exciting. You've done this study. What's the plan moving forward?
So we've got a lot of plans going on here in my lab now. We've got a major effort in trying to do non-genotoxic conditioning using antibody-based conditioning. So that's one of the things that I think will open it up even more. I think triosulfan is good and it's well tolerated, and I think it's worth moving forward that platform. I think there's opportunity in the transplant related realm, just the transplant aspects. And I have a graduate student who's working on that very hard, and we've got some interesting results there. So that's that. The other thing that we're doing is a phase 2, hopefully bringing that forward in India. It'll be a phase 2/3, which I think we will accrue very quickly, and I think we'll do that clinical trial fairly rapidly once we get somebody in India that knows how to make vector. And we needed to do technology transfer, and that didn't go as smoothly as I had hoped. But we're still on a reasonable pace to get some vector made this year or sooner. So I think the phase 2 is going to go forward in India. And then in the US, Expression Therapeutics is trying to do a trial using a second generation vector, and they're partnering with different people to try and bring that forward.
For hemophilia A.
For hemophilia A.
All right. Well, that's
Yep.
exciting.
Yep.
Trent, that's phenomenal. People can keep up with you. They can track the company Expression and keep up with your work coming out of Emory University. Are you on LinkedIn, or is there a way people can find you or anything?
LinkedIn, and if you can't find me, it's something's wrong. I'm everywhere. So
all right, he's everywhere.
Yeah, Emory is good.
Emory's good. All right, well, keep enjoying the Smoky Mountains, and thanks for taking time to share this story with us. And we wish you all the best in the future.
Not a problem. Thanks for inviting me.
Okay, Trent.
Talk to you later.
Take
Bye-bye.
care.